Hello students, so welcome back. Today we are going to discuss another concept from principles of inheritance and variation. In previous video we are discussed about linkage and the types of linkage. Now today we are going to discuss about if genes get inherited together, then how do we explain the law of independent assortment? So I am repeating the question as well as heading. If genes get inherited together, then how do we explain the law of independent assortment? Let us study an experiment done by Batson and Punnett. They made dihybrid crosses like Mendel did. How Mendel did. In the same manner, they did the dihybrid crosses. For that, they are chooses. Already you know very well dihybrid crosses is nothing but to considering the two pairs of characters, contrasting characters, two pairs of contrasting characters, contrasting characters. So now here the one first character, blue flower color, it was dominant. It is represented with capital B. Red flower color was recessive. So dominant color is blue, recessive color is red. And pollen size. Elongated pollen grains are dominant. Round pollen grains are recessive, small l and capital L. So dominant is elongated pollen grains, recessive is round pollen grains. These are the characters considered to carry the dihybrid crosses like Mendel. So let us discuss a cross between blue flower and elongated pollens and red flowers and round pollens. Blue flower elongated pollens are dominant and red flower round pollen grains are recessive. So the first cross made between these two, that is the cross between the dominant, that is capital B, capital B, capital L, that is a blue flower elongated pollen grains is crossed with is crossed with red flower with round pollen grains. So this is the first cross made between that. So then what is the gametes are produced here? The gamete produced is capital B, capital L. And here the gamete is produced, that is small b, small l. So during fertilization, these two gametes are fused together to form capital B, small b, capital L, small l. So this we are consider as F1 generation. This is F1 generation. F1 generation, and all this F1 generation is blue flowers and elongated pollens. So these are all blue flowers with elongated elongated pollen grains or pollens. So all these F1 generation plants are, so these F1 generation plants are also called as hybrids. All these hybrids are blue flowers and elongated pollens. That is the first result they got. So when they crossed these blue flowers elongated pollen plants with the red flowers and round pollen grain plants, then the first generation, F1 generation, all are blue flowers and elongated pollens. And these F1 generation, all the plants are in heterozygous condition. Heterozygous condition. So now after the getting this F1 generation, they made a test cross. So already we have studied that the test cross. How we did the test cross is defined as the cross the cross between the cross between F1 hybrids and homozygous recessive parent. Homozygous recessive parent. So all the F1 individual genotypes are capital B, small b, capital L, small l. This is the F1 generation genotype is crossed with homozygous recessive, that is small b, small b, small l, small l. So now here this F1, here it is heterozygous condition. That's why it produces total 4 gametes. That is capital B, capital L, capital B, small l, cap small small b, capital L, small b, small l are the 4 different gametes are formed from this plant. And here it is homozygous, that's why it produce only one type of gamete, that is small b, small l. This is the only gamete, all the gametes are having this, but there is a possibility of gametes, the 4 different types of gametes are formed. So as a result, what happens when you crossed this capital B, if you cross this one with this gamete, then it gives capital B, small b, capital L, small l. Then take this one and again capital B, small b, small l, small l. And this one, take this one, small b, small b, capital L, small l. And this one, small b, small b, small l, small l. So now this represent blue flowers, blue flowers and elongated pollen grains, elongated pollen grains. And this one, here it is capital B, that means blue flowers. All both are small l's, that's why round pollen grains. So blue flowers, blue flowers and round pollen grains. And this one small b. So small B indicates it is a recessive red flower. Red flower. And here capital L is the dominant. So that's why elongated pollen grains. Elongated pollen grains. And here both are recessive. That means red flower and round pollen grains. So if you compare out of these four individual, one is blue flowers out of four with elongated pollen grain, and one is blue flowers with round out of four, and one flower is red flower with elongated out of four. And then here a red flower out of four, one is a red flower and round pollen grains. So that means each individual forms 25% in the test cross. It is the test cross only. So when you conduct the test cross for this F1 individuals and homozygous recessive, 25% blue flowers with elongated pollen grains, 25% blue flowers with round pollen grains, 25% red flower with elongated pollen grains, and 25% red flowers with round pollen grains are formed. That means all the phenotypes are formed, four phenotypes are formed in equal proportions. So previous slides we are explained that blue flowers with elongated pollen grains crossed with red flowers with round pollen grains. So that is applied the dihybrid crosses which is done by the Mendel. So now on that same concept, we need to apply linked genes. So by the concept of linked genes, how this crosses will be the same cross. So now we are taken blue flowers with elongated pollen grains are crossed with plants with red flowers with round pollen grains. When we cross this cross, we are applied dihybrid cross which is done by Mendel. So law of independent assortment by we are explained based on the law of independent assortment. Now let's the same cross we need to explain by the concept of linked genes. If the genes for blue flowers and elongated pollens are present on the same chromosome, they should remain together and should be inherited together. In that case, they should give only two types of phenotypes. They should not be produce recombinants or there is no recombinants. I am repeating the statement again here. Blue flowers and elongated pollens are present together on the same chromosome. They are remain together and they are inherited together. Then only two types of phenotypes are produced. There is no recombinants. So here what are the recombinants we are observed in dihybrid cross? There are two types of recombinants we can observe. That is blue flowers with round pollen grain. This is, and another one is red flower with elongated pollen grain. These are the two recombinants we will observe in the dihybrid cross, or maybe independent assortment. These are recombinants we are observed during independent assortment. But in this linkage concept, what we are saying, these genes for these blue flowers and elongated pollens present on same chromosome, they should remain together and should inherit it together. That means there is no crossing over during meiosis. As a result, only two types of phenotypes are formed. The results should get by the concept of linkage genes are. So what we are saying, so we need to take here test cross only, test cross individuals only. So what are the test cross individuals? That is heterozygous, heterozygous, heterozygous blue and heterozygous elongated pollen grains crossed with homozygous recessive red and round pollen grains. What we are saying here, the blue flowers and elongated pollen grains are linked together on the same chromosome. These both are present on the same chromosome, and they are inherited together. There is no change. As a result, the 50% of individuals, 50% of individuals are 50%. So here they are present on the same individual. No, that's why it can produce only two types of gametes. One is capital B, capital L, and another one is small b, small l. So it is present on. So blue flowers and elongated pollen grains are present together on the same chromosome. Suppose if it is a one chromosome, the same chromosome consists capital B, capital L. And similarly, if it is a paired one, another chromosome consists small b, small l. Now, during gamete formation, this will goes to the one gamete and this will goes to the another gamete. Only two kinds of gametes are formed. And here also same. So one chromosome having small b and small l, and another chromosome having small b and small l. So then each gamete getting small b, small l. So all the gametes are same here, that is small b, small l. So when this fuse together, these two, the cross occurs between these two, it results capital B, small b, capital L, small l. When cross occurs between these two, this and this, then it gives capital, sorry, small b, small b, small b, small l, small l. So two individuals are formed. In that, one is uh, one is blue and elongated pollen grains, and another one is red and round pollen grains. That is 50% of blue and elongated pollen grains, and 50% is red and round pollen grains. So that's why the proportion is 1:1 ratio. So we are discuss the cross between blue flowers and elongated pollen grains crossed with plants with red flowers and round pollen grains. So we are applied law of independent assortment, which is proposed by Mandal. We are applied, and in test cross we are find that 25% of parental kind, 25% of recombinant, and another 25% another recombinant, that is 25% blue and round pollen grains, blue flower and round pollen grains 25%, and red flowers elongated pollen grains 25%, and 25% of red and round flower round pollen grains. And when we are applied by concept of linkage, then we are find only two types of phenotypes like a parental kind, that is 50% of individuals offsprings are blue flowers with elongated pollen grains, and 50% of flowers are red and round pollen grains. But we are discussed this cross is done by Batson and Punnett. Okay. But the result that Batson and Punnett got were as given below, which did not fit in any category of law of independent assortment or maybe linkage. It is not belongs to any category what we are discussed in previous just before slides, what we are discussed. In one category, 25 each equal proportion of recombinants and parental kinds are equally appeared in the test cross, whereas in linkage only two types of parental kind, that is only 50% and 50% of parental types only appeared in second category. But whatever the result got by this Batson and Punnett are not fit to any category of one or maybe two. The first category is law of independent assortment. Second category is linkage. It is not fit to the any of that category. So now what that explains now. So now in this category, when they are crossed, this F1 individual, that is capital B, capital L, small l, is crossed with small b, small b, small l, small l. So it produce a different kinds. That is, this cross produces total four kinds. In test cross, we will get the four individuals. So one is a blue flowers with elongated pollens. It is appears 43.7%. And second type of individuals, that is again blue flowers, blue flowers with round pollen grains, round pollen, it is only 6.3%. And another individuals, that is flowers red flowers with elongated with elongated pollen grains, again it is 6.3%. And lastly, the red flowers with round pollen grains appear with 43.7%. So as per linkage, the parental kinds, that is red flowers with round pollen grains, blue flowers with elongated pollen grains, these are two are the parental kinds. So as per linkage, it need to come 50% each. But here the new recombinants are appear with less than 50%. So here the parental types were two parental types. It is 87.4%, the parental types. And the recombinants types were 12.6% is the result got by the Batson and Punnett. So this result is not belongs to the category what we are discussing first, law of independent assertment. In that all these four appear 25% each in first category. In second category, the parental types are occurs 50% each. 50% for uh, blue flowers and elongated pollen grains, and 50% for blue, that means red flowers and round pollen grains. This is in second category and this is in first category. But the result got by Batson and Punnett, it is not belongs to this any, it is appears in these percentages. So here in linkage, both together 100%, 50 plus 50. But whereas in this Batson and Punnett result, the both parental types only 87.4%, not 100%. And recombinant types are occurs 12.6%. But whereas in first category, recombinants are also 25%. But here only 12.6%. From these results, category 1, 2 and Batson and Punnett results. From these results, following conclusions can be drawn. Two genes did not get independently asserted as the recombinant kinds were less than 50%. They definitely showed a pattern towards the linked genes. The two gene did not get independently asserted, did not separated. As recombinant kinds were less than 50%, they definitely showed a pattern towards the linked genes. Now let's see that why the 50% is appears here. See, so linked genes remain together without crossing over. That we need to. That means no crossing over is occurred. That means linked genes. If you recollect the linked or complete linkage and incomplete linkage, if you refer that in previous video, we are given that types of linkage: complete linkage and incomplete linkage. In complete linkage, there is no crossing over. In that case, capital B, capital L are linked genes on one chromosome. Linked genes on one chromosome. So this is. And small b and small l are linked genes on other chromosome. Both chromosomes form a homologous pair. So these two chromosomes form as a pair. Okay. Now these chromosomes, they can give two types of gametes. So one gamete, when they are assert, when it form the gametes, so this combination will goes to the one gamete, that is BL, capital B and capital L will get in one gamete, and small b small l is goes to the another gamete. And in another parent, that is homozygous recessive, red and round pollen grains, that is both the chromosomes are having kept small b, small l. So as a result, only one type of gamete is formed. That gamete is BL, small b, small l. Then when the fuses is takes place, one is 50% is blue elongated and 50% is round red flower with round pollen grains. 1 is to 1. So that means here if genes are linked together, linkage is stronger when genes are very close. That means distance is less, linkage is more. When distance is less, crossing over is absent. Due to lack of this crossing over, there is no recombinant and directly 50% of parental kinds, each of 50% is formed. So a pair of genes is said to be linked if their recombinant frequency in a test cross is lower than expected value of 50%. That is the conclusion. Remember that here 50% of one parental character and 50% of another parental character. This is happened due to the linked genes and it is happened due to lack of chromosome. That means crossing over between the chromosomes. So in another conclusion, the linked genes did separate out and thus recombinants were formed in a smaller number. This was later explained by the phenomenon of crossing over, which occurs naturally during meiosis. So the linked genes did separate out and thus recombinants were formed. So let's see here. So in previous slide we discussed that is a complete linkage. Now here it is incomplete linkage. Here it is. So the linked genes are separate. When the linked genes are separate, when the distance is more between the linked genes, a distance is more and they is easy to separate. How they are separate? Through crossing over. So that is represented here. See. So same concept. So two chromosome, the chromosome, one chromosome having, so in a pair of, it is a one and second chromosome. These two, in this pair of chromosome, one chromosome have capital B, capital L, and another chromosome have small b, small l. So when crossing over is takes place, then there are four types of gametes are formed. Four types of gametes are formed. But in this crossing over, the percentages are different. So the gamete formed here, it is almost 47.7, 43.7. And the recombinant gamete, that is small b, capital L, capital B, small l, these are only 6.3%. So based on that, we can get a different proposition of individuals. So blue flowers with elongated pollen grains 43.7%, red and elongated pollen grains 6.3, and red, blue and round pollen grains 6.3%, red and round pollen grains are 43.7%. So when compared all these, the recombinant occurs only 12.6% and parental kinds are less than 100%. Two parental kinds is less than 100%. The conclusion. of Batson and Punnett experiment is given in a tabular form. So the phenotypes we saw in the explanation are blue and elongated pollen grains, blue and round pollen grains, red and elongated pollen grains, red and round pollen grains. Type of combination, that is blue and elongated is a parental type of combination. Red and round pollen grains are also parental kind of this one. So this is also a parental kind, and blue and round pollen grains are the recombinant type combination, and red and elongated pollen grains are also recombinant type of combination. So the Batson and Punnett observed frequency is parental frequency of blue and elongated pollen grain is 43.7%, and another parental combination red and round pollen grains are 43.7%, and blue and round pollen grains 6.3%, red and elongated is 6.3%. So what expected frequency is? Expected frequency for genes asserted independently. If genes asserted independently, the expected frequency is 25% each. 25% of blue and elongated pollen grains, 25% blue and round pollen grains, 25% red and elongated pollen grains, and 25% red and round pollen grains. And frequency, expected frequency, if genes are fully linked, that means there is no distance. When genes are fully linked, there is no recombination because of lack of crossing over. So that's why only parental combinations are formed. 50% blue and elongated pollen grains and 50% red and round pollen grains plants are produced. There is no formation of recombinants if genes are fully linked. So recombinants are formed when genes are not fully linked. That means incomplete linkage leads to formation of recombinants due to occurrence of crossing over. If they are completely linked, then there is no chance of crossing over. Recombinants are absent. Thank you, students. We will discuss another concept, that is crossing over, in the next part.